Hello everyone, this is Cassandra Owen from the Innovate, and I'm here basically to take you through the somatic death and the postmortem changes and their related conditions. We shall begin with the definition of what somatic death means. So somatic death basically means the cessation of life in the entire individual that follows the failure of the vital body functions, and these vital body functions include the circulatory, the respiratory, and the central nervous system. So this somatic death usually happens when the body is deprived of oxygen, and this is one of the intermediate causes of the death, which is in most cases called asphyxia. So this one will be accompanied or will result into respiratory distress. This respiratory distress will be characterized by the following factors, whereby the animal or the individual will be having forced respiration, and also the other thing, you will be having accelerated heart action. That's when the heart is pumping at a high rate because it needs to supply oxygen to the different body tissues to supply this oxygen that is lacking kind of. And also there will be a spasmodic quivering of the musculature, of the body musculature. The reason being due to the decreased oxygen and the accumulation of lactic acid within these muscles. So it will be the one that is bringing about this quivering or the contraction of these muscles that is not spontaneous, that is not in irregular rhythms. And then also we shall be having irregular respiratory movements because of still the failure of these vital functions that are concerning the respiratory system. So that is all basically about the somatic death. And before we go to the postmortem changes, you will be seeing that in somatic death, there is something, the animal, when it is undergoing into a process of dying, that is a dying state, it is now called a moribund. A moribund animal is an animal that is undergoing, which is in the dying state. So from there, now we go to the postmortem changes. So basically here we are to say what are postmortem changes. So they are simply series of changes that occur after the death of an individual. And if you were to see that these postmortem changes are just a result of autolysis, or what we call decomposition, postmortem decomposition, rather than some pathological changes or events or processes, so therefore postmortem changes are not lesions. So autolysis just means that it is the process by which the body or the cells release enzymes that are going to eat up these cells. So they release enzymes that eat up these cells themselves, and that is the process that is going to result into autolysis. And after autolysis, we shall be now seeing these postmortem changes. So then as you see that this autolysis will result into morphological changes in cells and tissues, and therefore pathologists will be depending on this to distinguish from cell degeneration, or what we call necrosis in most cases. Now, while these postmortem changes are many, but we shall be looking at the vital ones or the most important ones within the veterinary sector. And one of them is rigor mortis. So rigor mortis refers to the stiffening of muscles and joints after death. But this usually occurs in muscles that have been active in life. For example, the eyelids, the muscles of the joints, those ones that are joining the joints. So those muscles that have been active that the body utilizes every time, they're the ones that are going to be affected by rigor mortis. So they become stiff after death. Now, what are the causes? So there are basically two mechanisms that have been put forward to describe the mechanism of how rigor mortis comes about. One of them is the swelling of the colloid that is brought about by accumulated lactic acid. So this lactic acid that has been accumulated before the death of the animal is sometimes thought to cause the prevention of muscle relaxation, and therefore muscles will be stiffened after the death of the animal due to this lactic acid. And then the other one is the depletion of ATP. So we very know well that in muscles to contract, they need ATP to break the tropomyosin bond for muscles to relax. But if the ATP is not there, then these muscles will remain in a sustained contraction. In most cases, we call it tetanus. But in this case, it will be something that is tetanus that is prolonged. So then the other postmortem change is the liver motis, or what we call hypostatic congestion. So liver motis is just the settling of blood on the lower part of the body due to the gravity. So if the animal dies and maybe this animal falls down, the blood will settle on the lower side. That is where the animal is resting. So blood will go down on that side of the body. And this will result in reddening of tissues observed externally. So if you observe it externally, most especially in animals that have pale skins and also have sparse hairs like the way you see in pigs. And so then internally, you can observe it after maybe the postmortem itself, and you'll be seeing this redness in organs and tissues. But as you see here, an example of this pig. So this is what we mean. This pig, when it died, it was resting on this side. And then you see this reddening here like this. So in regions where you can't see the blood, that means there was compression here, and therefore blood was pushed to the other sides of the body. Yes. So then we go to the next one, which is called alva mortis. So alva mortis, basically, this is refers to this gradual cooling of the body after death. We know very well that when the animal dies, for example, in humans, that our normal body temperature is 37 degrees Celsius. But when you die, the dead body, when you touch it, it will appear as if it is very cold. So what causes this alva mortis? The responsibility of alva mortis is the cessation of all metabolic activities responsible for heat production. One of the primary things that cause heat production in the body is the metabolic activities that take place within our bodies or within the bodies of animals. So if the animal dies, that means literally these things are going to stop, and the resulting thing will be that they are going to result, the body is going to cool down because all the vital metabolic functions have ceased. Then we go to postmortem autolysis. So basically, this one I talked about it. Postmortem autolysis is the digestion of tissues by their own enzymes under aseptic conditions. Now, here is where cells will release after the death of the animal. That tissues, for example, you know very well that most of the cells have what we call lysosomes. And these lysosomes have hydrolytic enzymes within these cells. So in cases when the animal has died, most cells will tend to burst or get damaged. So when they get damaged, they are going to release these enzymes to the rest of the body. And when they release them, these enzymes are going to start eating up these other cells and tissues and organs. And what will be the appearance as what we see grossly? Tissues will become soft. When you touch the body of the animal, it will appear as if it is very soft. Then microscopically is what we are seeing here. Microscopically, we'll be seeing pachynosis. So if you were to look in some of these cells, pachynosis means that there is a darkening of the nucleus due to its condensation. So when you see that in some cells here, you'll be seeing that the nuclei are very dark staining. These blue ones, I can't zoom this picture, but you will zoom it and you will see it very well. So some of the nuclei here are densely condensed, and that shows pachynosis. Then And also we shall see the granular coagulation of the cytoplasm. So if you were to look like, for example, this cell here, you'll be seeing that the cytoplasm coagulated, like it came into one part like this. This is what we mean. Something like this. Yes. Or this, or this here. Yeah, this is a clear one. So that is it. Then the next one is hemoglobin inhibition. So hemoglobin inhibition refers to the red staining of tissues, especially the intima of the blood vessels. So intima means the inner walls of the blood vessels and also the heart, basically by hemoglobin released by the red blood cells as autolysis sets in. So we know that, as we have said, that one of the postmortem things that occur is the postmortem autolysis. But at first, as I said, that postmortem changes as a result of autolysis. So they occur after autolysis occurring within the body. Before the animal, after an animal dying, autolysis has to occur before it dies. And then when it dies, these are the changes we shall be seeing because of the one we call postmortem changes. Now, one of the cells that you have in the body are red blood cells, and they have hemoglobin. Hemoglobin, which is red in color. So when the red blood cells are destroyed, or they are damaged, and they have been broken down by these enzymes, they are the enzymes that are running within the body, eating up the tissues before the death of the animal. This hemoglobin will be deposited into other within the blood vessels and also the heart walls, and that is what will cause this hemoglobin inhibition. So these blood cells and the heart will take in this hemoglobin, what we call by inhibition, taking it in. Yeah. And then the other one is pseudomelanosis. Pseudomelanosis is just the blue-green to black discoloration of tissues, especially along the digestive system. So you see that in pseudomelanosis, what causes this? One of the things we have said that happens is the lysis of the red blood cells, or the breakdown of these erythrocytes. So when that occurs, they are going to release hemoglobin. And when hemoglobin is released, the resulting, then also in the body, we usually have putrefactive bacteria. So these putrefactive bacteria, they release a component that is called hydrogen sulfide. And this hydrogen sulfide is going to combine with the ion that comes from this hemoglobin, because hemoglobin will further be broken down to, and the ion within that was in the hemoglobin will combine with the hydrogen sulfide to form iron sulfide deposits. So these ones are the ones that will be deposited within tissues, and is going to cause this iron sulfide is usually black in color. So in most cases, you'll be seeing that in pseudomelanosis, they call it the darkening of tissues, if you find in some textbooks. So it's because of this iron sulfide. Yes. The other postmortem change that we have is postmortem emphysema. So postmortem emphysema is what sometimes we call bloating. Yeah, it can be called bloating. The accumulation of gases within the tissues, resulting into the increased size of tissues. So gas production by enteric bacteria, those are the bacteria within our GIT, resulting will continues unabated in postmortem period. And what will happen is that there will be distension of the entire GIT. And also, if the GIT expands more and more, in most cases, that will also cause another postmortem change, which is the displacement of the viscera, what we call organ displacement. Because of for example, if it is in our herbivores like the ruminants, if the rumen broadens so much, it will now push and squeeze or patch and make patches within or displace organs like the heart and the liver. Yeah. But if the more it enlarges beyond its elastic limit, the other result will be rupture. So it will burst. And then also it can lead to eversion of the rectal mucosa. So where you see that the animal died, but the intestines as if they are out behind it. And that is the rectum coming out, basically the rectal mucosa only. Yeah, evading out. So, and also many other changes that can occur within this thing. So from here itself, I'll talk about the intestinal displacement. Intestinal displacement, but we shall see it there. So which is here? The intestinal displacement, the cause of it is still the other one, the emphysema or the bloating. The gas that accumulates within some organs like basically the GIT organs, like the stomachs, the intestines, is going to lead to increased intra-abdominal pressure, which pushes some organs out of their position, as I've explained earlier on. And also the other cause can be the peristaltic movement in the intestine shortly after death. As these intestines go on moving, their peristaltic movement that they make can cause the results that we saw above, like the invasion of the rectum, and also still displacement of certain tissues, organs. Yeah. Then the other postmortem change is the lens opacity. So lens opacity is just where the lens becomes opaque and white. That is a postmortem. And what causes this is the partial freezing of the carcass, which usually we discussed earlier on. We said it is called the gradual cooling, which is the alpha mortis. Yes. So that one. And so in this case, the lens, what it differs from, so in most cases this can be confused with cataracts. But what differs in lens opacity is that here the lens reverts to normal transparency on warming. Yes. So that is the only way we can differentiate it from cataracts. It's found within certain animal species. Yeah. The other one is the mucosal sloughing. This is just the peeling of the mucosal surface of organs. And as some of the causes, as we say, can be the what? The other one, which is the peristaltic movement within the GI tissue after death. It can be due to the enzymes that have been released. Yeah, that is a mucosal sloughing. So the mucosa will be peeled. Yes. Then another postmortem change is edema, which is the accumulation of water fluid within the intestines or the extravascular space. Just water from other tissues will be entering into certain, basically the intestines or any other space that is found within the body, and you will see that the animal within that region where this water has accumulated, it will swell and will be soft. When you touch it, it will be soft and consistent. You press it and it depresses because it has water inside. Yeah. Unlike in the emphysema or bloating, whereby you can touch, that basically occurs within the stomach region. When you touch it, as you saw in the other picture, the animal is just, and when they cut it, it will be having an odor that is really not good. Yeah. I will dissect it. Then we have bile inhibition, which is the diffusion of bile into the adjacent tissues. And as this bile goes into these adjacent tissues, it is going to stain those tissues. But this especially happens to organs that are in contact or near the gallbladder where this bile is being stored. Yeah. So this one first appears first in the endocardium and the intim of large blood vessels. But eventually this colors surrounding tissues. Yeah, so it will first occupy the blood vessels that are within the endocardia and also the endocardium, and then it will spread to other tissues, but you can start from anywhere depending. Yes. Then the other postmortem change is the postmortem clotting. So postmortem clotting is just the clotting of blood which does not get attached to the walls of the blood vessels, unlike in the antemortem clot, or sometimes that we call thrombus, where it can attach on them. But this is a postmortem clot. It doesn't stick to the walls of the blood vessels. So this postmortem clot is divided into two layers. Only where we can understand this is a postmortem clot. One of them, the other layer, that is the upper layer, which is usually gelatinous. So it will be as if it is shiny. I think you've ever seen it. I didn't get you a picture here, but you'll be seeing. So that upper layer is called the chicken fat clot. Whereas the lower layer, which is usually reddish, so it is the layer where blood has concentrated most and has clotted. Then on the upper surface it will be like liquid. Yeah. So this lower layer is reddish and is called the current jelly. Yes. So this one, the postmortem clotting usually still occurs within blood vessels, but basically even within the heart, because that's where blood concentrates more. Yes. So you will find these postmortem clots basically within organs that are more supplied or that have been more vascularized in life. Yes. So there are then other postmortem changes. There are very many that I haven't talked about. But basically those are the major ones, as I said. So lastly, I will talk about the factors affecting the postmortem changes. One is the cause of death. So what caused the death of the animal will determine what postmortem changes you're going to diagnose within an animal. So did the animal die of maybe an accident? The animal die of poisoning? Did it die of like anything that can cause death? Yes. So the cause of disease will determine the postmortem changes or will affect the postmortem changes that you can observe. Then the ambient temperature. So ambient temperature, if it is very cold, like the one we saw in our rigor mortis, that is stiffening of muscles. If the animal has died in an environment that is very cold, this will lead to a prolonged rigor mortis period. So the stiffening of the muscles will occur for a long period of time. But if it is dead, for example, in areas of Karamoja, where the temperatures are very high, so it will affect that the rigor mortis cannot last for long. Yes, because this ambient temperature will cause the muscles to relax back again to normal. And then the body temperature, death will also affect. If the animal died when its body temperature is very high, still that will cause some postmortem changes not to be seen easily or to disappear easily. Still, like rigor mortis, I would use the same example. The nutritional status of the subject. If maybe this animal died of starvation, that means that some postmortem changes will be more prominent than others. Let me say that, so this can be related to the cause of disease. Then the other is the time elapsed after death. As for example, in rigor mortis. So this rigor mortis usually occurs it starts from 8 hours after death, but it can last to 20 to 30 hours after death. So if the animal has died for over 48 hours, that means that if you assess for rigor mortis, you will not be able to see it. And in presence of certain bacteria, as we saw that some of the putrefactive bacteria produce hydrogen sulfide, if they are there in abundance, that's when you will be seeing these changes, some we saw. Yes, so thank you so much. That's what I have for you. Let us meet in the next session. Please subscribe to our YouTube channel and you'll be finding such content. God bless you.